Okay, so although I said thank you a few moments ago, but we were left with some parts, like someone may question that from chemistry we know that when a bond is broken, it needs the input of energy, isn't it? Like when, for example, HCl, if this bond is broken, then we get, okay, then we get like hydrogen and chlorine, but for that we need to give in energy, isn't it? But according to biology says when ATP is broken down, it gives ADP, sorry, ADP plus phosphate, isn't it? ADP plus phosphate. So ADP plus phosphate, isn't it? So how come that when ATP is broken down, it releases energy? How? First of all, I want to say, listen, that ATP does not broken down. ATP does not break down all by itself. Like HCl is being broken down itself to H and Cl2 without the help of anything. So ATP does not break down itself. So what actually happens in this case? What actually happens in this case is that ATP reacts with water to form ADP plus phosphate, or plus phosphate inorganic, or PI. Okay, so now let's look that what is actually going on inside these reactions and why it is releasing energy. Okay, now let's go. Okay, so first let's look at the structure of adenosine triphosphate. Okay, well, so this is the overall structure of adenosine triphosphate. Now let's look at what's going on. So look around here, what we have this is the ribose sugar, and this is the adenine, and I think one extra phosphate is unfortunately added here, which should not be. Okay, look, just wait, I need to modify something. So O, then O, then OH. Okay, now look, so what happens is that, look, in this structure here are three phosphate group for which we are calling it as triphosphate. Now look at oxygen here. This oxygen has got negative charge, this oxygen has got negative charge, and this oxygen also has got a negative charge. Okay, so there is a repulsion between these two oxygen atoms. Okay, oxygen ions or oxide ions. Okay, so when these two ions are repelling each other, when these two ions are repelling each other, this is being repelled in this side, this is being repelled in that side. Okay, all right, so there is a repulsion force, and due to this repulsion force, there is a stress created on this bond, there is a stress which is created on this bond, for which these bonds become really unstable. Okay, so that means this bond this bond and this bond, they are really very unstable. Same case goes here. There is a repulsion between this oxygen and this oxygen, so they will try to repel each other apart, creating stress on this bond, which we call the phosphodiester bond. Alright, now what happens is that when it reacts with a water molecule, when it reacts with a water molecule, sorry, when it reacts with a water molecule, what happens is that this phosphodiester linkage gets broken down, and one of the OH group, the OH group gets attached, the OH group gets attached to the phosphate, and the H gets attached to the oxygen. Okay, so what happens is that this linkage gets broken down like this. So this linkage, the whole linkage gets broken down like this. I'm copying it and pasting it over here. So this linkage gets broken down, and sorry, so this linkage gets broken down, and look, H is added to this O, right? So this part is broken, and we get what? We get like this molecule H, this is OH, this is OH, this was O minus actually, and to this part we have got an OH attached. There is a double bond, and this is a phosphate group. So this is what we are getting. Okay, so here we have an O minus, sorry, O minus. Wait, what? O, then minus. Okay, so this is what happens when ATP is broken down. So the broken down product, that means this product, the whole compound, this whole compound is now ADP, because there are two phosphate groups attached, so it is now ADP, and we have got a overall phosphate group. Okay, so look, the linkage formed here is what we have got is an PO linkage, which is now quite unstable, quite stable. Then what we have is that we have here an OH bond, we have here an OH bond, I guess you can see it. Here is a new OH bond, which is quite stable and quite stronger, and this PO bond is now also stronger. Okay, so what we are getting is that weaker bonds here and here are broken down to form stronger bonds here. So when a weaker bond is formed, when a stronger bond is formed from the weaker bonds, then energy must have been released. In this way, when ATP is broken down, it releases energy. It's simply that bond breaking takes in energy, and bond forming gives out energy. So when this bond and this bond is broken, it takes in a very small amount of energy because the bonds are already unstable due to repulsion between the oxygen or oxide ions. But the bonds formed here are quite very stable and very strong, so a lot of energy is released. That means breakdown of ATP releases energy in this way. Okay, so hope you have got it. Okay, so in this way, when ATP is broken down, it releases energy. And now I will be discussing about anaerobic respiration. Okay, anaerobic respiration occurs in the absence of oxygen. So all glycolytic enzymes, all the enzymes of glycolysis, the glycolysis are present in the cytoplasm of both human cells and also bacterial cells or yeast cells, whatever it is. But the bacterial cells or the yeast cells, that also contain a dehydrogenase enzyme in their cytoplasm. But in our cytoplasm, there is no dehydrogenase enzyme, for which, if you remember the steps of glycolysis, sorry, I'm doing quite wrong. It's not dehydrogenase, it's decarboxylase. Our cytoplasm do not have any decarboxylase enzyme, but the decarboxylase enzyme is present in the mitochondria. So our cytoplasm do not have any decarboxylase enzyme. So in the glycolysis step, there was no decarboxylation reaction occurring. So if there is no oxygen present in the mitochondria, then no acetyl if no oxygen is present in the mitochondria, then the electron transport chain would totally stop, because the ultimate electron acceptor would be the oxygen present. Okay, so if the electron transport chain stops, then the Krebs cycle would not work, and if Krebs cycle would not work, then the link reaction is of no use, so link reaction would not occur. So what will only happen is the glycolysis reaction. But what we have is that we have some dehydrogenase enzyme, or the dehydrogenase enzyme, and also we have the NADH produced from glycolysis, which would carry out the reaction in the way like this. You remember the structure of pyruvic acid, the CH3 C. Okay, let me go but below. So CH3 C double bond O COOH. So in the process of production of pyruvic acid, just remember that for one pyruvic acid, one NADH was produced. So that NADH will again react with this group, and NADH is strong enough to reduce the ketone group present in the pyruvic acid, but it's not strong enough to reduce the carboxylic acid groups. So it acts like NaBH4, sodium borohydrate, that you have studied in AS chemistry. So the NADH2 would turn into NAD and reduce the ketone group to a secondary alcohol group, COOH, which is what we are calling this product, which is what we are calling as the lactic acid. Okay, so number of carbon atom remains the same because we don't have a decarboxylase enzyme in our cytoplasm. So you might think that the link reaction also occurs in the mitochondria. Okay, the link reaction also occurs in the mitochondria, and that acetyl CoA transports that the coenzyme A transports the acetyl group in the form of acetyl CoA into the mitochondrial matrix. Okay. So our cytoplasm do not have any decarboxylase enzyme, so directly the ketone group is reduced to secondary alcohol group, and the product we are getting is called the lactic acid. All right. Okay. But in the cytoplasm of bacteria and yeast, they contain the decarboxylase enzyme. So the reaction that happens in them is that CH3CO COOH is first okay, it is first you can say that it is first decarboxylated. The carbon dioxide, the COO group is released as carbon dioxide, and the H group becomes attached to this carbon that forms CH3COH, the ethanol. Okay, it's the ethanol, the aldehyde group. So this aldehyde group is then reduced by NADH2 NAD. So they're releasing hydrogen will become attached here, and aldehydes are reduced to alcohols, so this will get reduced to OH, which is the primary alcohol, and this is the ethanol. Okay, so that's why inside bacteria and yeast, the product of anaerobic respiration is ethanol, because they have the decarboxylase enzyme, for which the carboxyl group COO is eliminated as CO2, and the H gets attached to this oxygen, forming the aldehyde, the ethanol. Okay, so that's the anaerobic respiration in our cases. Okay, so that's all for today. Next day we will talk about respiratory quotient, and then adaptation for rice in wet conditions. Okay, and then we will go to start the direct gene technology. Okay, thank you.